great thing about him is that he loves to take complex subjects and make them simple so that we can all understand. So it's really my great pleasure and of the symposium to welcome and Dr. Dennis Drayna. Thank you.
Okay. Well, thank you for that lovely introduction, and thanks to everyone for coming, getting out on a rainy day. Today, I have the pleasure of talking about viruses as a general class of things, but I've picked a few that have been in the news a lot because in general, people tend to have more questions about these. I've picked some that are well known to, I guess, everybody. And then I picked one just because it's, I picked Ebola just because it's very dramatic. So we'll talk a little bit about Ebola. Okay, so we'll just, very good. So first, I need to get everybody here on the same page so we all understand what a virus is, okay? So viruses are a unique class of thing in the natural world. They cannot grow by themselves. They grow only when they're inside the cells of some living organism, okay? What they do is they get in there by one way or another and hijack the machinery that we all use in our cells to do everything we need to do to be a living organism. So they are, in that sense, they're strict parasites, okay? They only grow inside cells. They're typically very, very small. They're maybe 1/10 to 1/100th the size of the cells they infect. They were not visible in any sort of microscope until the electron microscope was invented in the 1950s. So they were a great unknown. And for many years, whether something was a virus or a bacteria, some infectious agent, was defined whether they could go through a very, very fine ceramic filter. Okay, that's all they had back in the 1800s. So viruses, being so small, could pass through a filter, and once they went through the other side, were still infectious, okay? So there were so-called filterable agents. Bacteria, which are very small, were still big enough that they would get caught in the filter and could therefore not be transmitted through a filter. Okay, viruses are typically extremely specific to what they infect. In fact, sometimes some viruses are so specific, they only infect one particular type of cell in the organism that they infect, okay? So they're exquisitely specific in sort of what they pose a danger to, all right? Now, people often ask, well, are viruses then alive or are they not alive? I like to think of them as alive sometimes when they're in the host and they're replicating themselves, often causing a disease, then they're alive. But when they're outside the host, they're such simple organisms, you can actually crystallize them. They'll form crystals just like table salt. And you can take crystallized viruses and put them in a jar and put them on the shelf, and they're just stable there indefinitely, okay? And then they really don't meet any definitions of being alive. So that's how I think of viruses. The important thing is that all classes of living things, everything you can see around you, has viruses that infect it. So animals, plants, fungi, even bacteria have viruses that infect them, okay? Now, these things come in an amazing variety of shapes and structures, okay? Here I've just listed 3 for an example. Try this one here. So this is a bacterial virus. It infects one particular type of bacteria. these things are known as bacteriophages because they can sort of consume bacteria. They infect them and multiply and then blow up the bacteria, and then they're out. So viruses, the progeny, can go on to infect other bacteria. So this is a bacteriophage called T4. And as you see, it has an extremely detailed, subtle structure. Let's see, if you've seen the movie The Matrix, the probe that goes out looking for them. Well, it was modeled kind of on these things. These things use these thin feet here to actually find their host. And then they plunk this thing down onto the surface of their host. And then this neck contracts. And they squirt their genes into the host. And that's how they infect bacteria. This is tobacco mosaic virus. It infects plants, mostly tobacco. It's transmitted by insects. So an insect bites one, takes up some virus from an infected one, bites another plant, and infects it. Extremely simple virus. This thing is actually a long helix. That's what this thing is, this structure with its genes in this tube, sort of in the central area down the middle. And that is this helix, this tube that you see here, is just a single protein repeated over and over and over and over and over and over and over again. So tobacco mosaic virus is an extremely simple virus. It only has a few genes, including one that codes for this thing that's just repeated over and over again. Okay. This is smallpox, which infects humans. It's actually one of the biggest viruses that we're susceptible to that infects humans. Smallpox is a big family. Perhaps you've heard about monkeypox, now called Mpox. Viruses get named now these days to avoid stigmatization. So now they're calling it Mpox. I don't want to stigmatize monkeys, even though this monkeypox was probably never, ever in monkeys. It was probably always been just a human virus in any case. These viruses are all kind of related, but there's some are small and some are medium and some are big in terms of the number of genes. Smallpox is one of the biggest of the family. It has something like 350 genes, which is enormous for a virus. Most viruses don't have or need that many genes because they use the cell to make everything they need to grow. Okay. So extremely broad morphology, just in general. Okay, so how do these things live? I think I've mentioned this before. First, there's an attachment process. Then they get in. It turns out this getting into the cell is a fairly complicated deal. And we're going to talk a little bit more about that when we talk about COVID. Okay. Then once they get in there, they uncoat and they make lots of copies of their outside shell and their genes, which are just indicated schematically here in red. Then they assemble themselves back up and they get out and they're on to infect the next generation of unfortunate hosts. They get out two ways. They can either just dissolve the host cell, and it just bursts open and off they go. So polio is a good example of this kind of virus. Or they can squeeze through the membrane. They can sort of bleb through. And in doing so, they actually acquire a little bit of this outer membrane that is the hosts. Okay, so, and that leads to something called an enveloped virus. And we're going to talk about enveloped viruses today. Influenza is a famous one. So, as a general rule, I'll just say this membrane that surrounds envelope viruses, it's not very tough. And most of those viruses, in fact, just spread through the air. They don't live long once they get out of the host. They're kind of fragile. And it's really respiratory. That is, lungs to lungs sort of transmission, which is typical of things like influenza. Non-envelope viruses are things like polio, which live in the sewage, okay? And they're often very tough viruses. And smallpox is an extremely tough virus. These things can be very hard to get rid of because they just live in the dirt. They hang around for a long period of time. Okay, so I just want to take a second here to to emphasize the differences between bacteria and viruses they're both microorganisms. They're both microorganisms that cause disease, okay? Bacteria are free living. They have thousands of genes that code for everything they need to live. The smallest free living bacteria has like 1,800 genes, but a typical bacteria will have a couple thousand genes. So they can do all the stuff they need to do to be a free living organism wherever it is they live. They live on the surface of your skin. They live in your intestinal tract. They live out in the dirt. They live on the leaves of a tree. They live everywhere. They are evolutionarily very far away from humans. And so their genes are very different from human genes, and they code for things that are very different from the things in us. And so therefore, you can find things, which we call antibiotics, that basically murder something in a bacteria, and they don't murder anything in us because they're so different from us all the way down to what we call the molecular level, okay? So viruses are, as I said, inert outside a living cell. They have only a few genes, just enough to take over the host cell machinery and make copies of themselves. They are, in general, not susceptible to antibiotics, since they mostly use the host, in our case, our functions. And antiviral compounds are very difficult to find, as you can imagine. Anything you find, since the bacteria is mostly using us, if you want to inhibit a bacteria, it's going to inhibit us, right? So it's very hard to find antivirals, although a few have been found, and we'll talk about those. So I'm not a physician, but I'll just make this nod to clinical medicine here. When an infectious disease physician is presented with a patient that has a novel infectious disease, it is not at all obvious whether that infectious disease is caused by a bacteria or a virus. In fact, it can be very confusing. My favorite example is Legionnaire's disease. Legionnaire's disease is caused by, it's on the list here. It's caused by a bacteria. Many world-famous infectious disease clinicians thought for a while that it was probably a virus, okay? The story here is that there was an American Legion convention at a hotel in Philadelphia. A whole bunch of people got a pneumonia. 76 of them died. It was really quite frightening, and people didn't know what the causative agent was. The reason a lot of people thought it was a virus is because they tried a bunch of antibiotics, all the common antibiotics, in these patients, and none of them worked. So they thought, well, it must be a virus. It turns out it is a bacterium, and it is susceptible to antibiotics, but just very sort of obscure antibiotics that are not widely used. But you can see there's a whole variety of things with all sorts of different clinical presentations that are caused by both bacteria and virus. So it can be hard to tell. So let's talk about the genes that are in viruses. I'm a geneticist. I was sort of trained as a viral geneticist initially at working on viruses for my PhD. And so first we're going to do the quick detour through what are genes. Okay, here we go. This is the so-called central dogma of molecular biology. This seemingly simple scheme here, was basically figured out in the second half of the 20th century. And in doing so, there were probably 25 Nobel Prizes awarded to the people who figured this out, okay? The reference copy of our genetic information is DNA, okay? That's like the archive. DNA is very stable. I mean, you can get DNA out of like dinosaurs, you know? I mean, it's very tough molecule, and it's meant to be the repository where our genetic information, all genetic information sits, okay? It, however, is not the working version of our genes, okay? You can't turn on all the genes all the time, okay? Otherwise, I mean, what's the difference between the different cells in your body? Why is a liver cell so different than a skin cell, which is so different than a brain cell? Well, the reason is due to the which genes are turned on in that cell and which are turned off. That's the general phenomenon of developmental biology. And that business controls really how this archive is actually expressed to make all the different functions in all the different cells that we have, okay? It does it through a working copy of genetic information known as messenger RNA, mRNA, okay? So mRNA is what actually, through a process known as translation, makes proteins, and proteins do structure and function, okay? So they structure keratin, the protein of your hair, crystalline, the protein of the lens of your eye, okay? These are all proteins, and they're what do the actual functions in our cells. The functions of life are done by proteins, not only structures, but also all the chemical reactions that run, metabolism, all these things, right? That's all done by enzymes. Enzymes are all proteins, okay? So there's DNA undergoes transcription to make mRNA. mRNA undergoes translation to make proteins. And proteins you can sort of think of as operations, right, in an organization. All right. So, hello. So it turns out viruses, being the simple little things that they are, have found all different ways for their genetic information. Yes, there are DNA viruses. There are lots of them. Smallpox is a DNA virus. Chickenpox, herpes virus, all those are DNA viruses. But some viruses actually just cut straight to the chase and have as their genes messenger RNA. So polio just has a single messenger RNA. It makes one big long protein that's cut into 8 separate proteins, and that's poliovirus. That's it, okay? So when you think about this, all a virus really needs to do is to make messenger RNA. That's all it's got to do. Because if it can make messenger RNA, it can make proteins and take over, right? That's all it's got to do. So some viruses have one or more mRNA molecules as their sole genetic material. Other viruses have DNA, okay? There are some exotic forms out there. There's single-stranded DNA. There's double-stranded RNA viruses. And then there's one sneaky strategy that I'll mention. There's a group of viruses that are actually fairly common viruses of mammals called retroviruses. Why are they called retroviruses? Their genes in the virus, the little particle, is messenger RNA, okay? So they're ready to go, right? All they got to do is get in, and they're off and running. But they've decided they're going to do more. They actually make a process called reverse transcription. It's called reverse transcription somewhere here. Hello. A process known as reverse transcription that actually takes the messenger RNA, that's the genome of the virus, and reads it backwards. That's why it's called reverse transcription into DNA. And the DNA goes into our DNA. It becomes incorporated into our chromosomes. This is what HIV does. Well, once it's part of your own DNA, that's not good because you may be able to wipe out the virus, but there's still a copy of its genes in our own DNA. So it's not going anywhere. And so that strategy has been exploited by a number of viruses, just collectively referred to as retroviruses. Okay, so I say that we have antibiotics against bacteria. We don't much have antibiotics against viruses because they're so simple and they use all our machinery, right? But we have a few. So these are known as antivirals. They can be thought of as antibiotics against viruses. They can be, of course, difficult to develop because they use the host, our own cellular machinery, and anything that inhibits that is not going to be good for us. So there are a few unique weak points that have been found and exploited. We have a couple. So there's this 30 drug called amantadine that works some for influenza. We have protease inhibitors. A protease is a protein that cleaves other proteins, okay? It's a type of an enzyme that just clips other things. And a lot of viruses carry their own protease for various reasons I'll mention. And the drugs against HIV, the treatment of HIV has been revolutionized by the availability of antivirals, antiretroviral therapy, right? And it's because the HIV virus contains a protease, and the pharmaceutical industry is very good at making protease inhibitors. So that is actually what has allowed people to be alive with HIV, is they're constantly taking a protease inhibitor, okay? And then there's acyclovir, the herpes viruses, for instance, there's a few antivirals, and these things, herpes viruses are DNA viruses, and they have their own enzyme that copies their DNA once they get in the cell. They don't rely on the enzyme we use to copy our DNA. They bring their own, okay? And it has some slightly different characteristics, and one of them is it is susceptible to fake out. You can make something now called acyclovir that looks like a component of DNA, but doesn't, and gets incorporated into the viral DNA by the virus enzyme that copies its DNA, right? But it doesn't work. It basically puts a letter of the genetic alphabet in that it's like the letter G, but once it gets in there, the DNA is dead. And so that works for herpes virus. All of these have to be administered early in the course of disease, often in the first day or two. And the problem is drug resistance, especially for the RNA viruses. DNA viruses have mechanisms to protect the DNA. DNA is the archive material, right? It has all sorts of things that make sure it stays intact. If there's an error or a mutation, there are things that survey DNA and fix it, okay? RNA doesn't have that. So RNA viruses just throw off mutations all the time, and we're going to talk about that shortly. Okay, so I'll just stop here because that's the end of the general introduction to viruses, in case people might have questions.
We'll get you a mic,
Yeah.
so hold on.
So I have to wait until people have a mic so that,
We're
because
getting
this is
there.
being recorded so we can hear.
I'm curious about the someone who is living with HIV, managed many years, has children. Talking about the impact on DNA
Mhm.
and the medications, lifelong
Right.
medications. Could you just talk about the
Right.
genetic impact
Right,
downstream?
It's a very severe genetic impact. Many people, many infants who are born from HIV positive people, but treated, they still get a copy of the virus in their genome. And so they still have to be on antiretroviral therapy. Yeah. Now, I should tell you that there is a technique, I talked about this at the Oakmont Futures Club, called CRISPR, invented at Berkeley, California, actually, that can modify genes in place. Okay, when the genes are in the cell, it can modify them. And there's just been some clinical trials. It's just very experimental because CRISPR is a little dangerous because it makes breaks in the DNA when it's making its modifications. And breaks in your DNA, the cell doesn't like this. This is a bad thing. It's the same thing radiation does, makes breaks in your DNA. And so people have been very slow with CRISPR therapies, but it looks like CRISPR therapies may, knock on wood, be able to actually look through the entire genome of an HIV infected person, find the HIV, boom, and kill it. So it's very promising, but it's it's kind of a scary strategy. So people are taking it a little slow. Okay.
We we hear so much about how DNA is, people recognize that criminal. Does the virus that gets into the DNA, wouldn't that change the DNA so it wouldn't be as predictive as?
Right. It wouldn't accept. So humans have a lot of variation in their DNA. Every one of us here, unless there are two identical twins in the room, every one of us here has different DNA than everyone else. But those differences are less than 1% of our DNA. 99% of your DNA and my DNA is exactly the same. And yours and yours and yours and yours, everyone. Okay. These DNA forensic methods rely on examining that 1% that differs between people. And it turns out when HIV goes in, it just goes in a few places, and most of that is invisible to these methods that are used in forensic DNA technologies. There's one difference between any two people about every 1,000 letters. And since our total genome is about 3 billion letters long, okay, there's 3 million differences between you and me. And so there's plenty of differences to use to identify different people, or you don't have to worry about HIV being one over here and maybe one over there, okay?
Well, I think I just realized I've made a mistake. I was thinking that chicken pox, almost all of us have had chicken pox. And is it the herpes virus part of that family?
It is. Chickenpox is caused by a pox virus, and chickenpox has a twist clinically. Most all of us, myself included, have recovered naturally from chickenpox, but it's not gone. It lives in nerve cells. It hides out in nerve cells. Nerve cells are kind of resistant to immune surveillance. They're kind of protected because the body doesn't want your immune system attacking your nervous system. So there are viruses, and chickenpox is one of them, that can hide in your nerve cells, and unfortunately, under the right circumstances that are not fully understood yet, it can come back. But when it comes back as shingles. And I don't know how many of you have had shingles. I had shingles. It's not fun, right? But it comes back in a sector. I had mine here, but sometimes people just get it in one quadrant. And that's because it's innervated by, when it's hiding out in the spinal nerves that just innervate one part of your body. And that's the part that ends up with the symptoms of shingles. Okay, let's go on here, because I want to start talking a little bit about influenza, if we can. Influenza, it's a big deal in the world of human viruses. Has been for a long time, probably always will be. It's a pretty simple virus. It only has 8 genes. It's a so-called plus-stranded mRNA virus. So in other words, each of these 8 segments of RNA is a messenger RNA. It's a so-called segmented genome virus. Okay, they're not all in one piece, like for instance polio. It's membrane bound, so it blebs out of its host cell. It's transmitted mostly by respiratory transmission. It can lead to fatal pneumonia. It still kills lots of people. I mean, people forget that in modern day and age with modern medicine, it still kills tens of thousands of people a year just in the United States, and we have great health care. It's a serious killer. Here's a picture of it. These membrane-bound viruses can kind of sometimes look like blobs because they just, they don't have a clear, simple outline. Okay, so like all viruses, it enters the cell, it uncoats, it releases the 8 messenger RNAs, which get translated into 8 different proteins. They're also replicated into many copies of each one, and they get assembled and packaged, one of each of the 8 types into each particle. Particles bud out through the cell membrane to escape and go on to infect the next cell or the next person. Now, here's the thing about segmented genome viruses. Because these 8 genes each represents its own separate messenger RNA, if a cell gets infected with 2 different influenza viruses at the same time, and this happens, especially in agricultural settings, and especially in Asia, where people often live in kind of close proximity to their livestock, the resulting progeny viruses contain a mix and match set of whole complete genes that can come from either of the 2 different viruses. And here's, this is my, see, this is why I was a genetics major and not an art major. Okay, so here we have the 2 different viruses, one with all of its 8 genes in blue, and then the different kind with all of its 8 genes in red. If they go in, they just get packaged into random combinations of blue and red, 8 each new virus. And what comes out could be very big changes. So wholesale differences in the progeny from either of the 2 parents. And that leads to something known as antigenic shift. Okay, so the viruses are often classified by what kind of, whether an antibody that recognizes one recognizes another one or doesn't recognize another one. So is it immunologically distinguishable, or is it apparently the same? So that's antigenic shift, and it is contrasted with something known as antigenic drift, which is small successive changes. Okay, so most viruses that do not have a segmented genome just pick up a mutation here, they pick up a mutation there. One of them, they pick up mutations all the time. Most of them probably don't help the virus, but once in a while, one is actually a little better. And if you want to talk about it, and that leads to something called antigenic drift. So slowly but surely you get changes. We now have an example of antigenic drift on steroids, which is COVID. COVID is the world champion, or has been the world champion at antigenic drift, because it's an RNA virus, it just picks up lots of mutations, and it has a completely naive human population to just sweep through. Okay, all right. So antigenic shift is not the gradual accumulation of small mutations over and over again. Okay, so influenza has 2 major proteins on its outside. One is called neuraminidase and just called N, and the other is called hemagglutinin. It makes red blood cells stick together, hemagglutinin, and it's just called H. Because these 2 molecules face the cells of the host, they are the main target of the host defenses. And different types of H and N proteins have been identified and named just in the order of discovery. Okay, so the first ones were the 1918 worldwide influenza pandemic. It's sometimes called the Spanish flu. It almost certainly did not start in Spain, and killed tens of millions of people. It was an enormous, enormous pandemic. Killed mostly the young. That's why it was the 1918 pandemic was so scary. It killed people in their 20s and 30s. That thing was H1 and N1. They just look, this is a type of the H, and this is the type of the N. We're just going to call that 1. Okay. A new one came in the Hong Kong flu, which they then called H2 and N2. Then in the 60s, H3N2 began to appear. Earlier this year, we're now up to H8N3. That virus just appeared last spring. Okay. So how does all this affect the flu vaccine? Well, how do they deal with this? Each year in the spring, the World Health Organization assembles a group of experts. And these guys, these people are, they're an extremely impressive group of people. They represent leading academic physicians, infectious disease specialists. They represent molecular biologists. They represent the pharmaceutical industry, vaccine manufacturers. They represent epidemiologists, statisticians are on this panel. So it's a very high-powered group. And they decide, based on the best information available to them, what the most likely major types of influenza are going to be circulating the following fall. Okay? They provide guidance to governments and vaccine manufacturers, and production, which is now done in chicken eggs. That's how the vaccine is made. And that's why when you get your flu shot, they ask you, do you have an allergy to eggs? Because the virus actually grows in embryonic little chickens, fertilized chicken eggs. And it's an amazing process. Millions of eggs. And they're growing influenza, and they do it every year. They start from scratch with whatever strain is predicted to be the worrisome one for the coming flu season. Okay? Sometimes the World Health Organization's best guess prediction turns out to be correct, and sometimes not so much. I think 2 years ago, protection was like at 17%. I mean, it was just like, oh, maybe not such a good guess. But then again, it's not really their fault because influenza can change quite drastically in a fairly short period of time. And a new antigenic shift, quite a new virus, can emerge in a relatively short period of time. Okay, this year's flu shot, I just neglected to mention there's two types of influenza. There's A and B. B is typically considered a little less severe than A, but B kills people all the time. This year is a combination of the H1N1, H3N2, and so-called B Victoria. That's Victoria, Australia, where that strain first came from. Now maybe this is like Hinduism. You know, the problem with Hinduism is the continual cycle of death and rebirth. And, you know, and it's infinite, right? Is there some way we can avoid this and just come up with a so-called universal influenza vaccine? So, for instance, as an example, measles, right? Measles is a RNA virus. Its genome is one single messenger RNA molecule. You can make a vaccine against measles, and measles undergoes antigenic drift. It's picking up mutations here and there. But the vaccine recognizes something that if the measles changes it, it's dead. The measles absolutely has to have it to infect human cells. And that's what the vaccine is directed against. Okay? So measles can undergo antigenic drift, but it can't change what the vaccine is directed to. And so the vaccine, one dose of the vaccine, you have lifelong protection. Well, there must be something that influenza uses that's the same thing. Influenza has to have something that to infect human cells that can't change. Otherwise, it can't infect human cells, right? So people are working on it. And there are some really good candidates out there. I can talk, so I knew about some of them when I was at the NIH. And we can talk about those in the questions if you want. But there is hope for a universal influenza vaccine, and with any luck, it'll be one shot and you're done for your life. Okay, so that is an encouraging one. There's a lot of problems that I have just glossed over, but I think it's quite promising. Okay. On to COVID-19. Is there anybody here who is not sick of hearing about COVID? Okay. The name comes from coronavirus disease of 2019. It was chosen to avoid stigmatization. The first reports were of an acute severe respiratory disease leading to potentially fatal pneumonia in and around a city in China, single city in China called Wuhan. Okay. It was quickly established that this was caused by a coronavirus. Okay, so in general, coronaviruses, which I'll tell you about in a second, were well known from animal diseases. But in general, humans, there were no coronaviruses that caused any sort of significant human disease. Now, there were a couple that had emerged a few years earlier. There was something called SARS and something called MERS. This stands for Middle East Respiratory Syndrome, and this one stands for something acute respiratory syndrome. Both of these were in Eurasia. Both of them had emerged with sort of a little scary. Killed people. And so it looked like, and these were, there was nothing, there were no human coronaviruses that caused disease before this. So there was no vaccine. There were no antivirals. In fact, those diseases, if anybody here knows this fellow, Tony Fauci, he's right. So I'll just say, I know Tony quite well from my time at the NIH, and what you see on television is just the regular Tony Fauci. There's no acting. So Tony is a remarkable guy because not only is he an extremely skilled clinician, infectious disease clinician, but he has very heavyweight, sophisticated training in epidemiology, which is basically a field of mathematics. So Tony, remarkable guy. He pointed out that the reason these two diseases didn't sweep the world was just dumb luck. And that I can explain that as follows. It turns out for those diseases, humans are not infectious. They can't infect other people until after the symptoms appear. In many diseases, you are infectious before you're symptomatic, not for those two. And so because of that, you could use old-fashioned quarantine to just shut those people up, and you could stop the spread. And that's what they did, and it worked. So, but it was just dumb luck. As Tony Fauci said, the world missed the bullet on those two. Okay. So by the time COVID and its threat were recognized, it had already spread worldwide. To date, there are more than 500 million cases, more than 7 million people have died. Mostly kills the elderly. That's why, for instance, it hasn't had a high mortality rate in Africa, because Africa has extremely, in general, young population, growing fast, and so you get lots of young people in there. They don't die much from COVID. 1.1 million people in the United States have died of COVID, and that's with the vaccine. So this is fairly sobering. So the question here is, was the virus manufactured? We don't know, but I'm going to address that in 2 slides. So here's the virus. Corona, the Latin word for crown. So when you look at it, it sort of has this crown-like structure. It's a membrane-bound virus. It exhibits a spike protein on its surface. Here's the spike. Pretty clear what you see. And as I said before, most of these things have been animal viruses. The fancy word for this is zoonosis. These things have been zoonoses. So veterinarians worry about them. Human physicians, not so much. It's a very simple virus, the virus itself, the thing that spreads from person to person. 4 different proteins. That's all it's got. Just 4 different things. One of them it's an RNA virus. So it has a protein that wraps around the RNA called nucleoprotein or N protein. It's got an envelope protein. It's something that it organizes the membrane that it blebs through, okay, to escape. And that is done by the membrane protein N. And then there's just the spike protein and the envelope protein. That's kind of it. And all the vaccines are directed against the spike protein. Okay, if you've been vaccinated, you have raised antibodies to S. So if it's such a simple virus, let's see, I want to make sure. Yeah, the viral genome is a single large RNA molecule. It's 30,000 letters long. So that's big. To give you some idea, what's a good example? 30. Influenza is like, I think, a total of 6,000 letters long, total. The virus genome acts as messenger RNA, and it encodes lots of genes, including a bunch of large proteins that are cleaved into smaller ones before they take off and do their thing. Okay. I'm sorry, I got this off this magic internet thing. Ignore this. This is just a diagram. What I want to show you here, this is the genome with the genes sort of blocked out in different colors. Here, I mentioned the matrix protein and the nucleoprotein, and let's see, where is spike? Oh, here's the spike protein. This purple thing is the gene that encodes the spike protein. But what are these things? ORF, open reading frame. It's just a bunch of letters in messenger RNA that can clearly code for something. We don't know anything about these things, and they're big. So there's lots of function. There's lots of things that this virus can do that we don't understand yet. And it's part of the reason that pathology is so difficult with this virus. Okay, so but people are working on it. We know the structures. We just haven't figured out what's called the pathophysiology. Okay, now COVID strains. I thought I would talk about a little bit of this. The variants appear by mutations, usually simple mutations, one at a time. This is antigenic drift, okay? And it's named, the resulting variants that come out and become apparent to the infectious disease community are named just according to the subsequent letters of the Greek alphabet. So alpha was the first one. It was identified in Britain. Beta was first seen in South Africa. Delta was the first one that looked like it was causing problems. I don't know if you guys remember this. This is now ancient history. But Delta was the first one that people were getting scared because it was killing people. Lambda first appeared in Peru. Omicron, a second variant of concern, first was seen in both South Africa and Botswana. It was very different than all the others, and it had many mutations. So how did this one arise? They think what might have happened is that a person who was immunologically suppressed, maybe because they had AIDS, untreated AIDS in Africa, got infected with COVID, and they couldn't clear the virus. So the virus just lived in them and lived in them and lived in them, and one mutation, then another mutation, then another and another and another, and finally it got out and off on its way. And by that point, it was very different. But that's the current idea of where Omicron comes from. And most of the variants out there now are sort of Omicron derived, okay, the things that we've got. And if you got your booster, I got my booster here, I don't know, 3 weeks ago. That one was, that one is sort of the most recent variant that's out there. It's a subvariant of Omicron, okay? Okay, now to the question, was it manufactured? The short answer is we don't know. The information is probably not as complete as we need to be able to say. Okay. However, I think there's some things are pretty clear. I think any reasonable person could agree that the first cases were in Wuhan, China. What is Wuhan, China? Wuhan, China happens to be the location of a national laboratory run by the Chinese government, the Chinese health authorities, that specialized in the study of coronaviruses. Most coronaviruses come from bats, okay? And in fact, the world's experts in coronaviruses were Chinese investigators working at the Wuhan Institute, okay? The fact that the first public cases and the first fatalities happen to also be in Wuhan can't just be a coincidence, in my personal opinion. I am not an epidemiologist. I'm just a virus guy. But the question is, what happened? Maybe I can talk a little bit later if people are interested. It is often reported in the media that the United States, in general, through Tony Fauci, funded this laboratory in Wuhan. And in fact, they did give them some money because there were no human coronaviruses. There are no coronaviruses really in the New World. There just aren't. So if you're going to be ready for some new epidemic to sweep across the world, you've got to know something about these viruses. You have to. So where is the only place in the world that actually has these viruses and has a lab that you can study them? Well, it happened to be in Wuhan, China, and the NIH did give them some money. Well, they stopped giving them money a while ago, and the reason why was because they flunked their audit. Well, they didn't just flunk. When the NIH gives out a research grant, they don't just hand you a wheelbarrow of money and say, have a nice day. These things are audited as you go, right? And they were having trouble getting the level of information they wanted from the grant recipients. And they finally said, we can't keep doing this. Well, this was a long time ago. And at that point, by that point, they had actually learned quite a bit about coronaviruses and what these things were. And to, I think, the Chinese credit, they wanted to be the first ones to sequence the genome of this new infectious agent, and they were, okay? And they dumped it out into the public domain immediately. Okay, they're all extremely, I think, admirable and I think very important. And ultimately, it was those sequences that were used by the mRNA vaccine companies, which I'll talk about, to make the vaccines that are most widely used today. Now, the earliest cases look like they had kind of a different mutation from any other known coronavirus. So they knew the sequence. There are many coronaviruses, as I say, most infect bats, but some infect other animals, and they had the sequence of these things. They could recognize different varieties, okay? But this one had something odd. It has a 12-letter insertion in the gene encoding the spike, okay? It's kind of a weird mutation. Now, all sorts of strange things happen, especially in RNA viruses, which throw off mutations all the time. But it created a site for furin cleavage. What is that? Well, furin is a protease, that it's an enzyme that cleaves other proteins, and it's on the outside of our cells. We all have furin on the outside of all the cells in our body. And it cleaves the COVID spike protein, and in doing so, it can now efficiently gain entry into human cells. So this was, people looked at this and said, aha, this has been weaponized COVID. Well, there's no evidence that it's been weaponized COVID. Weird mutations like this have happened before in viruses. Viruses, as I say, RNA viruses make lots of mutations all the time. And of course, when one works well, boy, it's ready to go, right? It can just sweep through the human population. So people forget how big virus numbers are. I mean, a human who's infected with a virus will contain billions, many billions of virus particles, huge numbers. Well, if you have many billions, some of them are going to be different. And just 1 in a billion, if it has an advantage over all the others, it's off and running, right? So that's the way viruses do it. They do it with numbers. It's just regular sort of evolutionary selection. Mendelian, according to the laws of evolution, it just, the one with the advantage takes over and wins. It just does. So this then gave rise to lots of studies. The US government sponsored 6, and they were very different studies about the origin of this virus. Some were studies done by epidemiologists, people who studied the occurrence and the distribution of disease. Some were studies by infectious disease people who treat people with infectious disease. Some were done by laboratory virologists, virus people, okay? All sorts of different studies that took different approaches to try and answer the question, okay? Did this come from the laboratory in Wuhan? And the answer is we still don't know because the Chinese are not releasing the information. But it does seem like an incredible coincidence that the only place in the world this thing emerged was in the same city, okay? Although the New York Times, I don't know whether you saw it, it was a huge investigative report. It was pages and pages of the New York Times documenting where the first initial 50 cases were known to have probably started from, and they came from this market, this animal market in Wuhan, where people eat all sorts of different things in China, and they go to the market and buy them live, right? And so the New York Times said, well, look, the overwhelming evidence is that it just hopped from an animal to a human in this closed space inside this market, right? And maybe that's true. It could very well be true. I will just say, 2 of the 6 government agencies suggested it was a lab leak. So at the moment, the government is not saying it was a lab leak. Okay, but 1/3 of their own studies says that it could well have been. So I think by now you probably have a guess at what my personal opinion is here, but we just don't know. The information just isn't there. If somebody engineered a furin cleavage site into this, so I worked at a company called Genentech. Maybe you've heard of it. It was a famous company. Genentech is short for genetic engineering technology. Okay, I have engineered lots of genes in my day, lots of genes in viruses. You can't do that without leaving fingerprints in the genome. There are things there. There's just no common way, especially in China, which is good, getting better, but doesn't have any special, unique technologies, or certainly didn't in 2019. So people have looked for fingerprints of genetic manipulation and they don't find them. So who knows? Who knows? It almost certainly, in my humble opinion, came from the lab. Yeah. Is my time up? We're almost there. COVID antivirals. There's Paxlovid, which is a combination of 2 drugs that were developed for other viruses. It sort of works. The problem is rebound. You get over the disease and then you stop taking the drug and boom, it comes back. COVID vaccines. I'm just going to talk a little bit about COVID vaccines because they matter. There are traditional approaches. China just took the spike protein, used regular old-fashioned genetic engineering. It's a very safe, hyper safe vaccine. It's just a single protein. It's not virus. It's not in any way infectious. They did reasonable clinical trials. It sort of seemed to work, and it doesn't work very well. Brazil had a huge resurgence. Brazil did the Chinese vaccine, and it didn't. China won't tell us very much about their epidemiology, so we don't know how well it worked in China, but we know how well the Chinese vaccine worked in Brazil, and it wasn't very good. Instead of injecting the virus, just make mRNA, the codes for the spike protein. If you put mRNA, it will be translated into protein. And this, I should just mention, it's recognized as foreign and produces an effective immune response, although maybe not lifelong. The problem is messenger RNA is a pretty potent molecule. The body, you can't just have mRNA floating around in your bloodstream. So the body has mechanisms to recognize it and get rid of it. So to make an effective messenger RNA vaccine, you had to find ways to modify messenger RNA so it still worked, but wasn't recognized by your immune system and wasn't immediately cleared away. Well, a couple of people working at Penn, University of Pennsylvania, Carrico and Perlman figured out a way to do that. And 8 weeks ago, they won the Nobel Prize for doing it. And that's why we have the vaccines. And the great thing about messenger RNA vaccines is you don't have to develop a huge manufacturing plant if you want to make a new version. These vaccines, these are chemically synthesized messenger RNA. It's just done in a chemical tank. And you just punch in different numbers. First I want A, and then I want G, and then I want G, and then I want, and you say, oh no, now I'm going to do A, G, A, right? And so that's all you have to do to make a new vaccine with a messenger RNA vaccine. It's a huge logistic benefit. Okay, what's the future? The virus is now endemic, meaning it's out there. It's going to continue to exist. Let's see, new variants will continue to emerge. It's difficult to predict how infectious and how virulent, that is, how nasty the new mutant strains will be. And in general, I'll just say viruses and their hosts tend to reach an equilibrium because the virus has to have the host around, otherwise it can't live, right? So if it just kills all the hosts, it's not good for the virus. So they generally reach an equilibrium, and the infections will just sort of go along at some much lower rate in the population. We can talk about Ebola if people want to talk about it. It's very dramatic disease, kills people in Africa all the time. It's a completely different virus from the ones I've talked about so far. Comes from probably from it's a zoonosis. Normally lives in animals. Bushmeat. People in Africa consume bushmeat. And it looks like that's how it has outbreaks. It's around. It's been around since the 1970s, Ebola. It has sporadic little outbreaks here and there, but sometimes you get a bigger one. And those can be very, as I say, very dramatic because they have very high mortality. I mean, people are right to be afraid of Ebola. To give you some idea of how dramatic these diseases can be, severe cases, people bleed out of their eyes. It's like, and that's eventually they die of hypovolemic shock. They just bleed everywhere. They're so-called hemorrhagic fever viruses. You get outrageous fevers, 105, and you're bleeding. 90% mortality. They're very rare, but they're there. They keep coming out. They're filoviruses, otherwise known as, there's two of them superimposed here. There's just a string. It's a very weird virus. Only has 7 different proteins. It has its own way of copying its RNA. It brings it along in the virus. It has its own polymerase. Just because I mentioned it, here's the virus. It only has these few genes, but guess what? Here are 2 furin cleavage sites naturally occurring in Ebola. So there you go. It's pretty big. It has some big genes, 11 different proteins. And Ebola control, well, quarantine is still the first one. It's transmitted by human to human contact. These bigger outbreaks become human to human. They're no longer animal to human. Now it's human to human by contact. And there are vaccines, but it's tough to get these things delivered into, difficult places in sub-Saharan Africa. People talk about antivirals. There are no antivirals that work for Ebola. People are talking about getting vaccines and stockpiling them in strategic places in Southern Africa. So it's mostly a West African disease at this point. So final thoughts on infectious disease. It is an ever-changing landscape. There will always be new ones. I guarantee, I can't tell you what the next strain of influenza is going to be. I can't tell you what the next mutant version of COVID is going to look like, but I can tell you there are going to be new infectious diseases because it's just the nature of the business. Antibiotics and vaccines have changed life enormously. My mother grew up in the pre-antibiotic era. She reminded me, someone would they'd get a boil or a strep throat and they'd die. There were no antibiotics, you just died. Okay, enormous change in human health and life. Now there's resistance to vaccines, to becoming vaccinated. If people want to talk about population resistance to the concept of being vaccinated, I'm happy to do so later, but in a sense, scientists are victims of their own success. People here have not seen, something truly frightening. I mean, even though COVID killed 1 million elderly people in the United States, yeah, we were all scared. We all wanted to just stay home, but this was nothing like influenza, which killed like 10 times as many people, and they were the young people, or something like the plague, right? I mean, killed 1/3 of Europe, right? Modern infectious disease has put that behind us all. We don't have that anymore. And because of that, people are not so afraid of extremely bad infectious diseases because medicine can solve that problem. Well, if you don't see the effects of that, it's easy to develop an opinion, well, I don't need and I don't want a vaccination. But I think there is tremendous hope for the future in infectious disease. We've eradicated smallpox. There is no more smallpox in the world. It is gone. Smallpox, I mean, I lived in the era when smallpox killed people all the time, and the ones that didn't kill, you were left disfigured for life. I mean, this was a nasty disease. It does not exist anymore. They destroyed the last laboratory stocks, I don't know, 20 years ago. There's no more smallpox. People are getting close on polio. I think there's only a few countries left in which there's paralytic polio anymore. There is vaccination resistance in some of those places, other logistic problems, but I think we're very close to curing polio. I went to grade school with kids, my classmates were wearing crutches because they'd had paralytic polio. I think many of us, the same experience. It's gone. Okay, so this is the NIH Vaccine Research Center. It's the building next to the place where I work, and there are a lot of really smart people who are not getting paid all that much money, who are working really hard. Lots and lots of 12-hour-day kind of folks work in that building. And yeah, they work on scary things, but I think there is a great deal of hope for infectious disease in the future because the NIH has invested in understanding basic biology for 50 years, and now we're seeing the fruits of that understanding of how biological systems work, and the fact that we could get a vaccine that effectively stopped COVID in a year, it's an amazing accomplishment. So that's all I have to say. Thank you all.
Thanks, Dennis. We're going to take a few questions before we leave off today. Lynn, you have somebody that wants to start off with?
I do have a question. Can you explain why infants and small children are not really susceptible to COVID?
We don't know why. It might have to do with coagulopathies. A lot of people seem to get a lot of blood clots, and it's something that old people are susceptible to, and young people are less susceptible to. And so maybe that's what it is, but we don't know.
This is a little bit off topic, I think. Yeah, what, I'm sorry? Okay, a little bit off topic, and no offense to anyone here, but do you have any information as to the actual effectiveness of masks in a population?
Yes, masks work both ways. That actually some of that, so it's been known for a long time that masks protect others. That is, the uninfected are protected when the infected people wear masks. That's been known for like 40 years. The question is, did masks protect you from getting infected? And those results actually for COVID, and those results just came out in the past couple of months. And if you wear an N95, your chances of getting infected yourself go down by like 90% or something. It was amazing effects, what we call an effect size.
Great. And you mentioned the Nobel Prize in this year.
2023, about 8 weeks ago, yeah.
So the vaccine that you got here,
Yeah.
was that created using that information or the old information?
I gotta do this. Forgive me. Yeah. This woman, so I got the vaccine here. The first ones I got were Moderna. The one I got here was Pfizer, BioNTech. BioNTech is a German biotech company, and Pfizer sort of almost bought the company, but they licensed BioNTech's technology. This woman right here is the president, CEO, and chief scientific officer of BioNTech. So it actually is it's her vaccine, all the way to commercialization. No, the vaccines, is that the same vaccine we were getting 3 years ago? The vaccines have changed. They have evolved with all the different strains of COVID. So if you've gotten vaccinated and then boosted, you got different ones. If you've been boosted more than once, you've gotten additional different ones. It's the same technology.
So do you think possibly that technology has anything to do with this? And 5G was just set up in Wuhan around the time all this stuff started.
By technology, do you mean cell phone communication type of technology? You mentioned 5G, which is a cell phone technology. Right. 5G is, so I am not a physicist, but I do know that 5G is very high frequency. Most cell phones, they start out at 2 gigahertz, and now they're 3 gigahertz, and 5 gigahertz. You can get lots more data with a high frequency signal, but the high frequency doesn't travel very far. That's why you need lots of antennas. That you can't be very far from a 5G antenna. And do I personally think 5G technology has anything to do with viruses? No. It's like cell phones and cancer and brain cancer. I think there's no relationship, but that's just me reading the data. Lots of things are possible, but that's my personal opinion.
In reference to the viruses that become part of the host DNA,
Yeah, retroviruses.
is
Yeah.
there any concern about virus DNA getting into the reproductive cells of any animals and thus that becoming part of the genome of, say, us?
Oh, sure. There's tremendous concern. Tasmanian devils. Do you know what a Tasmanian devil is? Yeah. They're all dying of a cancer, and it's a retrovirus that's in their genes, and it's passed on to their offspring. That's why Tasmanian devils are on the way to becoming extinct. Sure, absolutely.
I have a question. You mentioned that the Wuhan lab lost its funding because it flunked.
It failed
Failed.
to produce enough information for the audits that the NIH requires when it gives out funding. And I can tell you my personal opinion about what was going on, but it's just a personal opinion. The Chinese are extremely anxious to be seen as a modern country with modern science and modern technological advances, and they want to do it themselves. They want to be the ones. And so they are very secretive about a lot of the things they do in their labs because they're afraid that the more advanced Western countries, like the United States, are just going to take their information and waltz away with it. And so they are kind of reluctant in general to tell all. They tell a lot of stuff, but they're not willing to tell all. There are also things work a little differently in China. There's, China, yeah, there are, right, one has to be, I think, vigilant in lots of places, not just China. One has to be vigilant about cultural factors that lead to lack of full information, for want of a better term. And so I think, I think there's a multitude of reasons why we don't have all the data we would like from the Chinese labs. Personally, look, in Pirbright, England, so there is a lab, a high containment virus lab in Surrey, so south of London, in the town of Pirbright. And one of the things they work on is a very devastating veterinary disease called foot and mouth disease. Okay, it affects cattle. And the only way you can control it is by slaughter and burning. And it's like, well, the only place foot and mouth disease ever appeared in Western Europe was 1 mile from that laboratory. Right? And I've been to Pirbright. I've given a talk at Pirbright. They are careful. I mean, I had to get like fumigated with formaldehyde walking in and out. That I couldn't wear my clothes, my own clothes. I got some kind of interesting clothes from them. But you can't, and they do, it's completely contained facility, and foot and mouth disease still got out. And they had to slaughter tens of thousands of cattle in England to stop it. So even and the people at Pirbright are first-rate people. And if it could happen at Pirbright, it could happen anywhere, frankly. So that's what I kind of think about Wuhan.
This will need to be our last question.
There's some recent data that suggests that influenza vaccine may help prevent against Alzheimer's. It's population-based, but can you speculate a rationale?
Yes, this is a really great question, and it touches on something that just actually got reported in the local news from this guy at Stanford, Emmanuel Mignot. Emmanuel's a friend of mine. He is a very, very talented MD, PhD, and he works on the immune system in the brain. And he actually is sort of beginning to figure this out, and it's probably true. Alzheimer's is, how Alzheimer's causes disease is still not completely understood, but it's known that if you can, this stuff accumulates, amyloid plaques and tangles, and a protein called tau and other things, clumps of stuff that shouldn't be there, begin to accumulate inside the cells in your brain, okay? Well, it turns out that the immune system may actually be capable of recognizing these abnormal clumps and clearing them. And so I think actually this is very new stuff. And it's one of the most exciting things I personally have heard in biomedical science in the past couple of years, because in fact, if you could use an immunology approach to Alzheimer's disease or frontotemporal dementia, which is sort of in the same vague general class, it could be extremely important. I mean, it could be a huge breakthrough, but it's a little early. Yeah. Okay, I think that's it. If anybody else has other questions.
Oh, I'm sure
happy
people
to
have
take
many,
them.
many questions. We are going to be ending, excuse me, we will be ending our presentation. Dennis, you'll be here for a few minutes after? Maybe?
Of course.
You'll be here for a few minutes after if you'd like to come up to address them, but we'd like to thank Dennis for a wonderful, wonderful presentation.
Thank you.
And we hope to
Thank
see
you.
you here next week.
Thank
Thank you
you.
so much.